Swayam Prabha. Digital India. Educated India.
Welcome back to my course Industrial Biotechnology. Now in the last lectures I discussed that citric acid fermentation process, and I told you that citric acid is a secondary metabolites, and it is produced by using Aspergillus niger, and two type of fermentation process are used for citric acid production. One is surface fermentation, another is submerged fermentation, but in the industry mostly they use the submerged fermentation process, and we observe that media that is used for the cell growth and media that is used for the citric acid fermentation process is different, because in case of inoculum vessel we produce the cell mass. In case of production fermenter we go for this product formation, that is citric acid. Now we observe that two metals plays very important role, most important significant role, as for example iron and manganese, because iron is a cofactor for the enzyme aconitase, because if you allow the aconitase enzyme to be active, then you will get very less amount of citric acid in the fermentation media. So that is to be removed from the production media, and also manganese ion that antagonize that effect of phosphofructokinase. The citric phosphofructokinase has been inhibited by as the citric acid formation take place. So that inhibition effect can be antagonized due to the formation of ammonia, which is take place when magnesium ion presence in the fermentation media is quite less. So then we discussed about the different uses of citric acid and details of the downstream processing. Now today we want to discuss one very interesting fermentation process, that is lactic acid, largely used by the biochemical industries. And lactic acid does not require any kind of introduction, because lactic acid is largely used as a food preservative. We might be aware that I can give a typical example that milk is a essential food for our survival, because this content most of the essential amino acids and good lipids, that is cholesterol and all this, that is good for the human health. But the problem is that we cannot preserve the milk for longer period of time. Even you pasteurize, after 3-4 days you have to use it. So milk, protein and fat, which is very good for the human consumption, that is usually preserved in the form of cheese. Because how cheese is produced? Cheese is produced by using the curd making process, and during curd making process we produce basically the lactic acid, and lactic acid acts as a preservative. That is how your milk, protein and fat, because it has low moisture content, milk has very high moisture content, so a lot of pathogenic organism can grow very easily. But since the moisture content in cheese is very less, so your contaminants, that that pathogenic growth will be very less. Also salt concentration in the cheese also quite less. So lactic acid is very important fermentation process, and is a lot of use for different purpose. So it's a solid state, it's a white and water soluble. Usually it is because we know the Embden-Mayrov pathway. In the Embden-Mayrov pathway, glucose is converted to pyruvic acid. Now, this pyruvic acid has the formula CH3 CO COOH. Now, this in presence of NADH so hydrogen that it is reduced and it forms CH3 CHOH and COOH. This is what is written here. Here CHOH and COOH, that is what we written here. This is called lactic acid. This is lactic acid. So, mostly the lactic acid fermentation process, we produce lactic acid in the form of calcium lactate, because we use the calcium hydroxide as for controlling the pH in the fermentation media, and lactic acid is usually converted to calcium lactate. And lactic acid found primarily in the sour milk such as the yogurt, kefir and some cottage cheese. The casein in fermented milk is coagulated due to lactic acid, and lactic acid also is responsible for the flavor of sourdough bread. Also find application in the pharmaceutical industry. Particularly, I want to point out here the polylactic acid has tremendous potential in the pharmaceutical industry. Particularly, if we look at the during operation, particularly if we have when we have internal operation in the early days, we usually use the nylon thread for any kind of stitching purpose. Now, we use some kind of polylactic acid material with clips and other things, and with the help of that we avoid kind of using like the nylon thread. So, if you use nylon thread, again you have to open the system and take it out, but if you use this lactic acid, polylactic acid, that will absorb in the system. So, you do not have to take it out. That has tremendous potentiality in the pharmaceutical market. So, you have two forms of lactic acid, we have L-lactic acid, we have D-lactic acid. So, mostly we produce the L form of lactic acid. Now, if you look at the history of lactic acid, it is very interesting. It was isolated and identified by Seeley that as the principal acid of sour milk in 1780. Then lactic acid was first discovered as a fermentive product by Blundeau in 1847. Then it was investigated by Pasteur as one of the first microbial problems, and Schulz that demonstrated the presence of lactic acid bacteria in the yeast culture, particularly distilleries. Here I want to point out another very interesting thing that I shall discuss after discuss the Baker's fermentation process, then I shall discuss it there. In the Baker's fermentation process, purposefully we add lactic acid bacteria in the fermentation broth. Because why we add lactic acid in the fermentation broth? Because this lactic acid acts as a preservative for storing these yeast cells. During the same period, the Delbruck, this is another scientist that was endeavoring to determine the most favorable temperature for lactic acid fermentation in distilleries. Then he concluded that relatively high temperature, about 45 degrees centigrade, 44-45 degrees centigrade, is a high yield of lactic acid. Then 2006, global production of lactic acid reached about 275,000 tons with an average annual growth of 10%. So, this indicates the importance of lactic acid at the industrial sector. Now, lactic acid bacteria are diverse group of bacteria capable of producing lactic acid, because in the day to day life we use the curd, and curd contains also a lot of lactic acid bacteria. Here we have mentioned lactobacillus, streptococcus lactis, they are largely present in our curd. and found in cheese, yogurt and decomposing plant. It is a gram-positive and non-sporulating bacteria. The ability to produce lactic acid as a major metabolic end products of carbohydrate fermentation. They are non-respiratory, that anaerobic, but tolerate also aerated environment and can survive pH 3 to 6 and high ethanol concentration. So, here I want to just emphasize that this is a micro aerophilic fermentation process, because little air is required, because if you for this fermentation process also, but it is also anaerobically it can also grow. The lactobacilli can be divided into different genera such as the lactobacillus, leuconostoc, pediococcus, lactococcus and streptococcus. Now, I was talking about the potentiality of lactic acid in the different sectors. First, I told you already that in the medicine we have lot of use lactic acid. Then the food, we have food and feed industry, we have a lot of uses of lactic acid. I can give the example of, of preservation of vegetables. As for example, that cucumber and cabbage, that if we can store, if you dry it and put it in a polythene pouch, you can store it for longer period of time. But problem is that when you dry this cabbage and cucumber, due to Brownian reaction, this color will be brownish, because the yellow color will be lost. So, you can preserve, but the color will be lost, the natural color, that is the green color, will be lost. Now, question come, how you can preserve this vegetable in the natural form for a longer period of time, and this can be done by using this lactic acid bacteria. How? Just in a polythene pouch, I can tell you, this is a polythene pouch. Inside the polythene pouch, you can keep the shredded vegetables or shredded cabbages, and then use some salt, and then you close it. You can close it here with the help of rubber band or something like this. It will, as soon as you use some salt, some water will be secreted out from the vegetables, and this water, lactic acid bacteria will grow, and due to the production of lactic acid, this lactic acid bacteria automatically will preserve the vegetable for longer period of time. This is largely used in the food and feed industries, and chemistry also, it is quite important. It is used as a probiotic. Nowadays, lot of pharmaceutical industry, they marketed probiotic as a medicinal aid, because this is the good bacteria that is required for our digestive tract. So, they produce this bacteria and use as a marketed in the form of tablet. And starter culture for the dairy food and non-dairy foods is used as a starter culture. Then we have antimicrobial agent, your biopreservative. I already talked about the medicinal sector. I told you that pharmaceutical sector, that polylactic acid has tremendous potentiality. Then it can be used as a ingredients, that is the enzymes, vitamins, exopolysaccharides and low caloric sweetener. Now, if you look at the lactic acid production, is two type of fermentation process usually carried out for lactic acid fermentation process. One is called homofermentive process and heterofermentive process. Now, question comes, what is the homofermentive process? Homofermentive process, usually that it follows the Embden-Mayer pathway, where mostly we get the lactic acid as a product. But when you use the heterofermentive process, that it follows the pentose, it follows the pentose pathway. It follows the pentose phosphate pathways, and So, let me talk about the and what the in the heterofermentive process, the problem is that the product not only that lactic acid, with lactic acid you will get some other additional product, as for example ethanol and other things, carbon dioxide etc. will form in the fermentation broth. So, homofermentive process, how it forms, I told you this, it follows the EMP pathways, and EMP pathways we get the pyruvic acid, and I showed you here in the pyruvic acid, when you NADH, that is pyruvate, that NADH is converted to lactate, that this is how it is produced. Now, activity required, the lactic acid bacteria require the complex media for their growth. Now, here I want to point out that what do you mean by complex media and synthetic media? Complex media means that when the exact composition of the media is not well defined. As for example, when you use in the media like yeast extract, any kind of natural product, it is very difficult to define the composition of the media. But when you use any kind of pure chemicals, it is possible to define the exact composition of the media. Now, if the composition of media is well defined, we call it synthetic media, or when it is undefined, we call it complex media. So, lactic acid required complex media for their growth. Then they fail to grow in the media containing lack of amino acids. Then it utilize the B vitamins, and they are now produced commercially from corn sugar, molasses and whey. Whey is the very good raw materials. Whey contains very good amount of lactose, and it is a very good raw materials for the lactic acid production. And usually the yeast extract or malt extract is used, which contain all the amino acids required for the growth of lactic acid bacteria. Now, desirable characteristics of the lactic acid during the fermentation, you should require the lactic acid is one of the major products. Lactic acid bacteria may form components other than lactic acid, as for example 2,3-butanediol. It should act as a preservative. It should prevent the putrefaction changes that taking place in sauerkraut. I told you sauerkraut is largely available, so it prevents the putrefaction. Putrefaction means degradation of the proteins, and pickles and souring of milk. It should aid flavor development in the fermentation product. Characteristics flavor of the dairy products such as yogurt and cheese is due to the lactic acid bacteria. The importance of lactic acid bacteria addition of yeast cell, I already discussed. I also told you that purposefully we add the lactic acid bacteria in the yeast fermentation process, because it acts as a preservative. You see that it acts as a preservative. Most yeasts utilize the lactic acid as source of carbon. Most of the contamination to yeast is caused by the alkalinity of the media. To avoid undesirable bacteria grow, like putrefying bacteria, to prevent contamination in the fermentation process, lactic acid bacteria is used. So, aseptic fermentation is relux. In ancient age, large inoculums are used to prevent the contamination. It is used as acidulant to adjust the pH of the brine in the manufacture of pickled green olives, and to inhibit the development of butyric acid bacteria in the manufacture of yeast. Now, this is the differentiation between the homofermentation, the homofermentative bacteria and heterofermentative bacteria. As I pointed out that here mostly the product we have the lactic acid and carbon dioxide, and maybe small amount of volatile fatty acid is produced, and most of the bacteria use this fermentative process, that is Lactobacillus casei, Lactobacillus delbrueckii and Lactobacillus bulgaricus. Now, I want to point out here in the large scale application, mostly we use either Lactobacillus delbrueckii and Lactobacillus bulgaricus. And homofermentive process, they differ from homofermentive processes in the fact that it produces the ethanol, include ethanol, acetic acid and formic acid and carbon dioxide. It follows the pentose phosphate pathway, an organism that is Lactobacillus acidophilus that is used. Now, spoilage reaction, this lactic acid has some causes some kind of problem with this industry. As for example, that it acts as a spoilage in the beer, malt beverage, wine and distillery industry. They produce turbidity, off flavor and precipitates. In acetone butanol fermentation, lactic acid bacteria is undesirable. It uses sugar for their growth. In dairy industry, it can cause the souring in milk. In orange juice, lactic acid bacteria causes the spoilage because of the off flavor and souring. In sugar refinery, the leuconostoc mesenteroid can grow which forms a viscous sugar which may interfere the crystallization of sugar. This is also very important. In meat product, it can form turbidity and surface growth which is undesirable. So, lactic acid bacteria not only used for has all the advantages, but it has several disadvantages also. Now, if you compare that chemical and biological manufacturing of this lactic acid production, it is like this. Chemically, we produce from petrochemical resources. It produces acetaldehyde, addition of hydrocyanide, and catalyst. It produces the lactonitrile. Then hydrolysis with H2SO4, it poses the reciproc mixture of DL lactic acid. Now, biological process is very simple. We have renewable resources. I hope all of you understand what is called renewable resources. Renew means which will be available again and again. As for example, I want to tell you that any kind of agricultural product that is renewable, because we can reproduce again and again. So, as for example, if we talk about starch, if we talk about glucose, if we talk about lignocellulosic material, all are renewable resources because you can produce again and again. So, these renewable resources we can do the pre-treatment, then we produce the carbohydrate, soluble carbohydrate. This undergo the fermentation process, then we get in the fermentation broth, then through the purification process we get the pure L and D lactic acid. Now, carbohydrates suitable for the utilization, a large number of carbohydrate may be utilized for lactic acid production. The acid is generally produced glucose from glucose, sucrose and lactose. The starch, corn and potato especially may be hydrolyzed by enzyme or by acid prepared with sulfuric acid to maltose and glucose. Molasses and whey are generally low price sources of sugar for the fermentation, because we have already seen that cane molasses is used as a raw material for citric acid industry. Then sugar in maize is normally adjusted to a concentration of 5 to 20%. The oxygen relationship that bacteria, as I pointed out, used to produce lactic acid industrially are usually microaerophilic or anaerobic in nature. Microaerophilic means it required little bit of oxygen for their growth and metabolism. Also, mostly grow under anaerobic conditions. Small amount of oxygen is required for the conversion of lactic acid also. Now, pH, that is very important as far as lactic acid fermentation is concerned. Here I want to tell you that we have seen that in case of citric acid fermentation process, we allow the pH to drop down as low as possible, that favored the citric acid fermentation process. Otherwise, we will get the gluconic acid or oxalic acid in the fermentation broth. Now, here the pH control is required. The fermentation proceed between best when the pH on the acid side of neutrality, owing to the addition of calcium carbonate or calcium hydroxide or some other neutralizing agent in the fermentation mass, the pH tends to approach to the neutrality, that is 5.5, and 6.5 is usually find most suitable for lactic acid fermentation process. And since we are adding the calcium hydroxide, calcium carbonate, so mostly in the fermentation broth we get the calcium lactate as a product. Now, then we do the hydrolysis, acid hydrolysis to get the pure lactic acid. Now, growth factor of lactic acid, southern growth factor appears to be essential for certain lactic acid bacteria. Riboflavin acts as an activator. Nicotinic acid stimulated growth and acid formation. Some strain require biotin, pantothionic acid and nicotinic acid for their normal growth. Now, accessory nutrients, the yield and the fermentation time depends on the kind and quantity and combination of the accessory nutrient. They also supply the organic nitrogen and stimulatory substances. Good fermentation yield, short fermentation time results from the addition of accessory nutrients, that is such as the malt spout and stiff water that is used for this as a accessory nutrients in this fermentation process. Now, this lactic acid fermentation that usually, as I told you, that usually it takes place very high temperature, is usually 40 to 50 degree centigrade temperature, and it can use the different carbohydrate. It can based lactose. It can use lactose. It can use sugar. It can use glucose as a carbon source, and also the starch. If you want to use the starchy raw materials, which will have to hydrolyze to glucose or maltose, this can be used as a substrate for the lactic acid fermentation process. Now, lactic acid is carried out comparatively high temperature. Fermentation temperature depends on the organism used. As for example, in case I told you that these two bacteria largely used by the industry, Lactobacillus delbrueckii and Lactobacillus bulgaricus. Lactobacillus delbrueckii requires the temperature of 45 degrees centigrade or higher, and maybe that may be maintained, and Lactobacillus delbrueckii comparatively a little bit higher because 45 to 50 degree. So, Lactobacillus casei, Lactobacillus pentosus and Streptococcus lactis, they require relatively low temperature, and so this temperature usually we favor for our homemade card production. We know that in the home we produce lot of card, and during card making also this particular Lactobacillus casei, Streptococcus largely grow in the card. So, this required comparatively low temperature. But fermentation time is quite high, 1 to 6 days that is required, and yield is about 85 to 90% of the sugar that is used in the fermentation media. So, lactic acid. So, in conclusion, I want to tell that that lactic acid is considered very useful useful product in the industrial sector, because it has several applications, and particularly for food preservation it is largely used. I told you polylactic acid has large application in the pharmaceutical industries, plus it has other several applications, and this is usually produced by the lactic acid bacteria, and two bacteria plays very important role, what is called Lactobacillus delbrueckii and Lactobacillus bulgaricus, and they are mostly they are homofermative. The heterofermative organism also used for the lactic acid production, but homofermative mostly we get the lactic acid is the major product, and carbon dioxide beside little organic acids. So, I think in the next presentation I shall show you the downstream and upstream processing of the lactic acid fermentation process. Thank you very much.